All right, and without further ado, it's my pleasure to introduce Dr. Claire Willing, who's a postdoctoral scholar with Dr. Kabir Pei at Stanford University. And Claire's research is broadly focused on forest microbiomes and climate change. And I forgot to say she's in the Department of Biology at Stanford University. Previously, she was at Berkeley, and it was my pleasure to overlap with her as we were completing our PhDs together. And she was in the Environmental Science, Policy, and Management Department with Todd Dawson. You had, I just forgot about, you had another advisor, right, Claire? I don't want to.
I worked with Tom Friends as well, yeah.
Yes, I didn't want to miss anyone. Thank you for filling in on that. I realized as I was speaking, I forgot to get that detail. And her research seeks to understand how global change influences patterns of microbial community composition in forests and the functional consequences for forest health, which I'm sure she's going to talk to us a lot about tonight. And she uses interdisciplinary approaches, including molecular tools, ecological theory, and techniques in plant ecophysiology and stable isotope ecology. The goal of her research is to elucidate the importance of microbial communities in the resistance and resilience of forested ecosystems to global change. And she also has fun hobbies like soccer and backpacking, and just a pleasure to work with. And I'm so excited to hear what she's going to talk about. Welcome, Claire. We're so excited to have you.
Thanks so much. I'm going to go ahead and share my screen. So let's hope we don't have any Zoom faux pas. Okay. And let's just hide that. Okay, awesome. So as Rachel mentioned, I'm going to talk to you today about forest microbiomes and climate change. And I'm specifically going to be talking to you about microbial communities as mediators of forest health. So this is a photo of the Taylor Creek fire in Southern Oregon in 2018. And we all approach images like these with different lenses. The first thing that some people might see might be the burning trees. Others might be thinking about the legacy of colonial fire suppression, which resulted in the high severity of this fire. And I see these things too, but what I also see is the bridge that I used to drive on to get to work every day where I grew up. I come from a rural low-income community in Southern Oregon, and I grew up surrounded by forests. And while I've always been really excited about forests, it really wasn't until college that I started to learn about the tremendous complexity of forest systems that had been below my feet my entire life. Underneath the soil, fungi and bacteria help to decompose leaves and organic matter and aid in the liberation of important soil nutrients and help control soil carbon storage. These microbes also interact with roots and help to determine plant access to limiting soil nutrients, as well as to water. And these microbes can even aid in plant defense against pests and pathogens. In this way, forest microbes are really key axes of both plant and ecosystem health. As I've watched climate crises unfold in the forests around me, I've become fascinated with how forest microbial communities might be responding and how this could help to determine the capacity of these forests to adapt to climate change. Of the different plant microbe interactions that take place in the rooting zone, mycorrhizal relationships, or the relationships between plants and specific fungal mutualists, are some of the most fundamental. Before plants had evolved roots, these are the fungi that are thought to have led the transition of plants from water onto land and helped act as the roots for these plant hosts. Today, an estimated 80 to 90% of plants form relationships with mycorrhizal fungi. And the few species which do not form relationships with mycorrhizal fungi have actually evolved to not have these relationships, largely in extreme environments. When we refer to mycorrhizal fungi, this actually refers to a very diverse group of fungi across the fungal kingdom. But there are actually many different types of mycorrhizal fungi that exist. One particular group that you're going to be hearing a lot about in my talk today are arbuscular mycorrhizal fungi. And these form relationships with an estimated 85% or more of mycorrhizal plants. Here in this photo, you can see a picture of a root and this tiny little thread coming off of it, which is the Arbuscular mycorrhizal hyphae. And then the spores of that hyphae are alongside that thread of the fungal hyphae. And these fungi traverse the soil looking for nutrients and water, and then form relationships with plants to attain carbon compounds like sugars that are necessary for their growth. And as the fungi travel within their roots, they make little pit stops in the root cortical cells, these cells sort of on the outside part of roots. And that's where they fill up on sugars in exchange for providing the plant with nutrients. To perform this exchange, the fungi sort of poke into the cortical cells of roots, almost like you poke your finger into a balloon without puncturing it. And in this way, the fungi are able to form these sort of branching structures known as arbuscules, which increase the surface area of exchange between the plant and the fungus. In the coming decades, forests are going to need to adapt to severe climate change. And as these forests experience increasing severity and frequency of climatic disturbance, my research program is really focused on how microbes will help shape the adaptation of forests to climate change, really from the ground up. And just as studies of gut microbiomes have really revolutionized our understanding of human health, forest microbiomes have the capacity to revolutionize our understanding of forest health and their capacity to adapt to climate change. However, while microbiomes likely shape the adaptation of forests to climate change, we first need to understand the forest microbiome, who the microbes are, their effects on plant systems, and how they are shaped by climate. For my talk today, I'm going to start by sort of sharing with you some of my research about how climate shapes microbiomes. And next, I'm going to talk to you a bit about how forest systems are really embedded in this context of coupled human natural systems, where legacies of land management can impact forest structure and how that can influence forest microbiomes as well. I'm then going to talk to you a bit about my work looking at how forest microbiomes influence processes of plant ecology and how microbes can shape the response of plants to their environments, including to climatic disturbance. And once I've established some of the ways that my research has investigated the climatic and human impact on microbiomes, as well as the importance of microbiomes for forest systems, I'm going to give you a little sneak peek into how I'm sort of pulling all of these pieces together to tell you about how I'm thinking about the future of forests in a changing climate, really from the ground up. And my approach to this work really sits at the intersection of three major disciplines, and that is microbial ecology, plant community ecology, and plant ecophysiology. So throughout this talk, you're going to be seeing these three symbols, not necessarily all together at once, but you'll be seeing these different symbols indicating places where I'm drawing on tools and theories from these different disciplines. Throughout the talk, you'll also see citations found on the right-hand side of my slides, which refer to the work of others, which have inspired my research, and any relevant photo credits, as well as on the left-hand side, you'll find citations for my work. And for the purposes of this talk, I'm not going to be going into too much depth about the statistical analyses that I've used, but for those interested, those are going to be listed beneath each plot. So to begin, I really first needed to understand the ways in which climate could influence microbial communities. In general, we know that microbial communities change across climate and environmental gradients. But so far, we've been struggling to find unifying ideas or principles that allow us to make predictions because microbial communities are so complex and diverse. Additionally, we have to rely on molecular tools to identify shifts in microbial communities because unlike plants, we cannot visually detect and assess changes in microbial communities in the same way. Within the soil, bacterial communities are incredibly diverse, but work in experimental and agricultural systems and grasslands has actually shown that a particular group known as actinobacteria are especially enriched during conditions of water deficit. And this is likely because they have this thickened cell wall layer that helps protect them from stress associated with water deficit. I became really interested in this group because many species have actually been shown to improve plant growth during drought. So this is a plot here from work previously done. And on the x-axis, you can see Streptomyces, a type of actinobacteria, increasing in abundance in the root system of plants in this case. And on the y-axis, you can see plant species drought tolerance, which is actually just a metric of plant growth during drought. And so you can see that as Streptomyces abundance increases in roots, plants are able to grow more in conditions of water deficit. And while we often picture bacteria to look like these unicellular rod-shaped forms, bacteria actually exhibit different types of growth morphology. Some bacteria are unicellular and travel through the soil profile using flagella or other types of locomotion, while other bacteria are actually filamentous and form shapes almost like fungi. And in the actinobacterial group, there are actually many bacteria that form these filamentous forms. And just as plant growth forms can help predict water use strategies, bacterial growth forms might also exhibit differences in water tolerance. In this photo here, you can see the co-occurrence of trees, shrubs, and grasses, and intuitively, you might even be able to tell visually where you would expect water to occur, or like water availability to be higher. For this study, I wanted to see if different types of bacterial morphology, unicellular or filamentous forms of bacteria, might also correspond to water availability. So for this experiment, I sought out to test 3 specific hypotheses. The first was that water availability would be an important predictor of bacterial community composition in forests. The second was that there would be a relative enrichment of actinobacteria with increasing aridity. And then the last was that bacterial growth morphology would really help to predict microbial responses to water availability. And I chose to test these 3 hypotheses in the redwood forests because it sort of acts as a natural lab for gradients of water availability, where the northmost part of the redwood range receives about twice as much annual rainfall as the southern extent of the redwood range. So for this study, I used a nested approach and I sampled 8 plots spanning the gradient of water deficit throughout the redwood range. And I took soil cores, and denoted here in red spots around each tree. And I was able to sieve those soils and take a subset of the soil to measure soil chemistry, and then a subset of roots and soils to do DNA extractions, PCR and amplification of a specific region that helps us identify bacterial species. And then I was able to use bioinformatics and data analysis in order to test these hypotheses. So to test how bacteria responded to a range of climatic and soil chemistry factors, instead of using linear approaches like Mantell tests or multiple matrix regression, I decided to use a curvilinear approach, which is called generalized dissimilarity modeling, because I assumed that many of the responses of these bacteria to these factors would actually not be linear. And I've plotted the results from these analyses here, but I'm going to start by sort of just explaining how to interpret these plots, because I have a feeling most people haven't seen these types of plots before. So on the x-axis, I've standardized the different environmental factors that were significant in these models, and they are so that they could all be plotted on the same graph. So 0 represents the mean of each factor for the range that was measured in this study. On the y-axis, I've plotted communities' dissimilarity. So at 0, communities are identical. And as we move up the y-axis, microbial communities become more dissimilar. And in the soils, what we can see is that soil pH is really corresponding or predicting changes in the bacterial community. But the second most important factor is water availability. And soil pH has been long identified to be a very important predictor of shifts in bacterial community composition. But finding this gradient of water availability to be a second most important factor was really exciting. And we found similar patterns for microbial communities associated with roots, where soil pH was the most important predictor of shifts in the bacterial community, but that water availability also helped to predict those shifts. In testing our second hypothesis, we wanted to see if actinobacteria would really be enriched in cases of increasing aridity across a natural gradient of water availability. So I used linear mixed effects modeling to specifically test if actinobacteria were more relatively enriched during drier conditions as had been previously observed. So in this plot, along the x-axis, I've plotted again scaled water availability, where 0 represents the mean water availability across all sites, and then going out from there. And then along the y-axis, I've presented relative abundance of actinobacteria, where I've standardized sequence reads to an even sequence read depth, and then compared how the relative proportion of actinobacteria varied across this gradient of water availability. And what we can see is that the actinobacteria in roots and in soils became relatively more abundant as we moved to the drier part of the redwood range. And I was also able to demonstrate that of this group of actinobacteria that were enriched during drought, it was actually really filamentous bacteria that were making up the bulk of that enrichment, particularly in roots. And so on the top line, you can see still this plot of actinobacteria across the range of soil or across the range of water availability. And then this bottom yellow line is showing us the non-filamentous proportion of those actinobacteria. So the actinobacteria that were filamentous are actually making up that difference and really driving a lot of this pattern that we see. So one explanation for these findings is that while unicellular bacteria are unable to move through the soil profile during conditions of water deficit, filamentous bacteria are still able to access their important water resources that they need and persist even in conditions of water deficit. So in summary, I was able to establish that water availability is an important predictor of bacterial community composition in forest systems, and that actinobacteria are enriched in conditions of increasing aridity. And last, I was able to show that bacterial growth morphology helps predict microbial responses to water deficit. So just like we see transitions between plant biomes, such as between forests to shrublands or shrublands to grasslands with shifts in water availability, my work has really been highlighting similar transitions between microbial morphotypes. And understanding some of this basic biology is going to be really critical in helping us predict how microbes are going to respond to drier climates as a result of climate change-induced drought. So I've shown you a little bit about how climate can impact microbial communities. And now I'm going to tell you a little bit about some of my research investigating the effects of logging on forest structure and how that can shape forest microbiomes and may impact their ability to adapt to climate change. Deforestation related to resource extractive economies poses a really important threat to forest biodiversity across the globe. Additionally, severe climate change could result in further impacts on landscape structure, where, for instance, droughts, hurricanes, or fires could further fragment forests. Settler colonial logging practices through the 1900s have taken a particularly huge toll on redwood forests. In fact, as a result of extensive logging, only about 5% of the original old growth forest remains intact. However, there's really minimal information on the relationship between landscape structure and microbial communities. And in fact, for much of Western scientific history, People have thought that shifts in landscape structure really shouldn't matter for microbes because they're so small and could probably just disperse everywhere. More recently, however, studies have shown that dispersal limitation does indeed exist for microbes and is actually a very important predictor of their community composition. As I had mentioned earlier, however, plant and fungal partners have been co-evolving for millions of years. And particularly for fungi, there seems to be evidence of strong biotic filtering in plant roots. So while shifts in forest structure might result in different microbial communities in the soil, for example, it's possible that plants are able to conserve their microbiomes even as the environment around them is changing. Additionally, throughout my work in redwood forest, I noted these sort of strange swollen structures on redwood roots, and I began to wonder if maybe they were playing a role in compartmentalizing important root microbes, especially where the pool of soil fungi might be more diminished, for instance, due to the effects of habitat fragmentation. So for this study, I sought out to test how settler colonial induced habitat fragmentation might impact redwood forest microbiomes. And specifically, I wanted to see if habitat fragmentation or reduced redwood cover could correspond to shifts in the fungal community and soils, reducing potentially the richness or changing the composition of those communities. I also wanted to see if fungal community structure and redwood roots might be sort of decoupled from these patterns as a result of strong biotic filtering. And last, I wanted to see if compartmentalization in these structures might actually be one way that roots are able to sort of facilitate further biotic filtering of the fungal community and help maintain their relationships with important symbionts. So in this abstract image here, red represents redwood forests and green represents other types of habitats. Extensive logging can have really two important impacts in forest structure. First, it can result in habitat fragmentation, reducing the connectivity of forest. Second, it can result in a sort of overall loss of habitat area. In this study, I sampled roots and soils of mature trees, again in this nested design, using 100 by 100 meter plots across a gradient of landscape habitat fragmentation. To quantify remaining redwood habitat and habitat fragmentation in the forest community as a metric of forest land use, I established these 10-kilometer radius buffer zones around each plot, which is the distance at which dispersal limitation becomes important for fungi. So that basically means that the pool of fungi available to the trees in my plots was largely limited to this 10-kilometer buffer zone. Then using these buffer zones, I was able to determine metrics of redwood cover and habitat fragmentation using the National Land Cover Dataset for California from the USGS. And then back in my lab, I was able to use similar methods as I described for my previous bacteria study, except that I used fungal-specific ITS2 primers instead of bacterial primers. So I amplified the fungal part of the DNA instead of the bacterial part. And what I found is that forest structure did indeed predict soil fungal community richness. So on the x-axis here, we have edge density. This is a metric for habitat fragmentation that was identified for each buffer zone. On the y-axis, we have the Shannon Diversity Index, which is used as a metric of fungal diversity. And what we see is that while there's a lot of variation in fungal diversity within a site, we can still detect a signal of landscape structure on forest microbiomes. And this is consistent with what we typically would predict for macrobes, even though people had other expectations for microbial communities. So essentially what we see is that as forest habitats become more fragmented, we get a decline in the richness or diversity of these soil fungal communities. And similarly to the study I did before, I used generalized dissimilarity modeling and backwards model selection to determine the most parsimonious models to essentially predict how these communities or the factors associated with transitions in these redwood fungal communities. And I want to point out that the sampling took place across the same gradient of water availability that I'd used for my previous study. And I used these same climatic variables in these models, but that fungi really didn't respond to shifts in water availability like bacteria. And I think that this is really because the bulk of these fungi, the most dominant groups, have these filamentous forms and are probably more resistant to shifts in water availability, just like we saw with the bacterial community. And so for this plot, I've plotted geographic distance on the bottom x-axis and redwood percent cover on the top axis, and then community dissimilarity on the y-axis. And so again, 0 is identical soil fungal communities. And as we move up that y-axis, the communities become more dissimilar. And what we find is that geographic distance between samples is really the best predictor of changes in the soil fungal community, but that redwood forest cover is the second most important predictor. And so in this way, forest structure really does help to predict soil fungal community structure. But what about the roots? So similarly to the plots that I just showed you, we have edge density along the x-axis here. And this was, again, used as the metric of habitat fragmentation. And then on the y-axis, I've plotted Shannon diversity for the fungal community associated with redwood roots. And importantly, what we find is that none of the metrics of landscape diversity predicted soil fungal or root fungal community diversity. And so in this way, root communities seem to be sort of decoupled from these patterns of landscape heterogeneity that shift the soil fungal community. And here again, I used generalized dissimilarity modeling for the root associated fungal community. And again, we find that geographic distance is an important predictor of shifts in the root community, but that actually soil pH is the second most important predictor of shifts in root-associated fungal communities, and that the structure of forest is not actually a great predictor of changes in root-associated fungal communities. And the last hypothesis we had was that these structures on redwood roots might actually help compartmentalize key fungal community members and help facilitate some of this biotic filtering or the maintenance of these fungal communities across these gradients of soil or of forest structure. And I visualize the fungal community here across roots, soils, and these structures using non-metric multi-dimensional scaling, which is just a sort of fancy statistical tool, but essentially each point is going to represent a given fungal community. And the closer these fungal communities are, the more similar the structure of those communities are. So the more similar taxa that exist and the more similar the overlap of those communities essentially are. And what we see is that root-associated fungal communities and the communities found in these structures are quite separate from the light blue communities of the soil, but that these root and structure communities are also distinct from each other. And even more exciting was that we found that these novel root structures actually served as domiciles for arbuscular mycorrhizal fungi. So on the x-axis here, I've plotted rhizonode, root, or soil, so structures, root, or soil. And on the y-axis, we have the relative abundance of AMF, or arbuscular mycorrhizal fungi. And what we can see is that the arbuscular mycorrhizal fungal community is really, really enriched in these structures. And because of their important role in compartmentalizing this really key group of fungi, I decided to give these structures a name, which is rhizonodes, as they're sort of these nodes of mycorrhizal activity within the rhizosphere. So we found that landscape heterogeneity really influences fungal community composition in soil, both in terms of the number of species and the composition of those species that are present. Conversely, we found that whereas root communities are indeed influenced by the soil community, they're actually somewhat resistant to shifts in landscape heterogeneity and are mainly shaped by soil pH. Additionally, we find that compartmentalization of the fungal community in rhizonode structures really further facilitates the maintenance of these redwood fungal symbiont communities. And we know that landscape structure is going to continue to be impacted by extractive resource economies and by climatic disturbances such as fire, drought, and hurricanes. And my research is really highlighting that these processes will potentially lead to a decline in soil fungal diversity, but that root-associated fungal communities might actually show some resistance to these shifts in these communities. But it's really important to note too that while this is potentially good news for trees, there will also likely be tipping points or thresholds after which trees may no longer be able to sort of access or maintain these important symbionts. So I've now shown you some of the ways that there are really strong effects of both climate and landscape structure on microbial communities, but I haven't told you what this means for shifts, what these shifts might mean for forests. So now I'm going to dive in and talk to you a little bit about some of my recent work that I'm really excited about, exploring how microbes can affect plant competition and coexistence. Forests are complex, multi-species assemblages where empirically we know that these species form stable coexistence. However, very few studies have actually been able to identify stable coexistence in plant ecology. And one reason for this might be that we've often removed plants from their environmental context for these studies. And while we often think of plants as individuals, trees are actually really part of forest systems, which includes their microbiomes, and plants are never found in nature without these microbes. So for this study, I specifically wanted to test if microbes might actually be a really key axis of plant coexistence. And to investigate this, I've been working in a relatively simple system in Point Reyes, California, where plant and microbial systems have been really well studied. And this has enabled me to essentially manipulate the microbial communities associated with some of the dominant flora and use them in a greenhouse study to investigate how these different pieces, the different components of these microbial communities might contribute to stable coexistence across the landscape. And I've talked to you a little bit about arbuscular mycorrhizal fungi, or AMF, but what I haven't really told you is that there are different types of mycorrhizal fungi that are host specific. So a given tree is typically going to form a certain type of mycorrhizal association, and it will not form associations with other types of mycorrhizal fungi. In this system, Baccharis is associated with arbuscular mycorrhizal fungi, and pine is associated with a distinct type of mycorrhizae known as ectomycorrhizal fungi. And for this study, I grew seedlings of both Baccharis and bishop pine, either alone or with 1, 4, or 8 competitors of either the same species or the competitor species. I also used 4 different mycorrhizal treatments, which you can see here, where I included no mycorrhizae, just ectomycorrhizal fungi, ecto and arbuscular mycorrhizal fungi, or just arbuscular mycorrhizal fungi. And what we find is that there's positive growth responses of plants to their host-specific microbes. So starting with Baccharis, we find that when AMF are present, they grow larger. By contrast, we find that there's actually even a slightly parasitic effect of AMF on pine. And when we add EMF, plant growth increases somewhat. And when only EMF are present, pines grow the largest. And ultimately, what we can see here is that when plants have their host-specific fungi, they're able to grow larger due to increased photosynthetic rates. However, this doesn't tell us what's going to happen when these plants are growing on the landscape together. So now we're going to look at what happens when we grow these plants in competition. I'm going to start by showing you just one plot first. And this is a plot of Baccharis as the focal plant grown with AMF. And what we can see is that on this x-axis, we've plotted number of competitors. And on the y-axis is the biomass, or the weight, essentially, of the focal plant Baccharis. And what we can see is that in the red here, the red line declines much more rapidly. And this is Baccharis growth when grown against other Baccharis. This blue line is showing the growth of Baccharis when grown against pine. And what we can see is that when AMF are present, Baccharis becomes more self-limiting, where this red line has a steeper slope than the blue line. And when we put this all together across the different treatments, you can use the differences between these slopes and intercepts to calculate niche and fitness differences across our different mycorrhizal treatments. I'm not going to explain the mathematical background of how these were calculated, but if folks want to email me, I'm happy to set up times to talk about that. And essentially, what we're able to derive from that is these values of both niche and fitness differences. And there's a lot going on in this plot, so I'm going to start by sort of explaining each of these axes independently first. So on the y-axis, we've plotted fitness differences between the plants, and that is essentially the degree to which one plant is able to grow compared to the other plant. And positive values above 1 indicate that pine is able to grow bigger. And by contrast, values below 1 indicate that Baccharis is able to grow bigger. This x-axis is showing resource use overlap. And positive values indicate self-limitation. And negative values indicate that these species are limiting each other more. So putting it all together, we can see that where fitness ratios are much above or below 1, we expect either pine to be able to invade Baccharis or Baccharis to be able to invade pine. Where fitness ratios are relatively smaller and plants experience more self-limitation, meaning Baccharis versus Baccharis or pine versus pine, species are predicted to coexist. Where fitness ratios are much smaller, plants where fitness ratios are equivalently small and where plants limit each other more, we expect to find that the plant that gets to that site first will be able to successfully establish, and we call this a priority effect. And what we saw in our experiment is that when no mycorrhizal fungi are present, pine is able to invade Baccharis. Similarly, when ectomycorrhizal fungi are present, pine is still able to invade Baccharis. But when we add arbuscular mycorrhizal fungi to that treatment, it pulls the system closer towards coexistence. And similarly, when only arbuscular mycorrhizal fungi are present, we predict that these two species, Baccharis and bishop pine, are able to coexist. So in building these analytical tools and identifying this important link between microbes and landscape scale dynamics of plant assembly, my research is really opening up some important inlets and avenues of future research, enabling us to ask how climatic disturbance, which results in shifts in microbial communities or in plant physiology, will potentially impact landscape scale stability of forested ecosystems. But microbes are also going to be important for forest trees' response to climate change. While I don't have time to go into this work today, I do want to just make a note that in addition to investigating how mycorrhizal fungi impact processes of plant establishment and coexistence, I've also been conducting research to better understand some of the pathways by which these fungi might help regulate really important plant hormones, especially those associated with stomatal closure, or the closure of the pores on leaves in response to water deficit. And mycorrhizal fungi and plants have been co-evolving for 450 million years or more. And consequently, there's very complex interplay between these fungi and their plant host, which will have really important consequences for response of forests to climate change. So ultimately, I've shown you how microbes are influenced by both climate and human land management, as well as how these microbes are critical parts of plant competition and coexistence, and potentially plant physiology, ecophysiology as well. So now I'm going to just briefly tell you a little bit about where my research is headed in the coming years. And specifically, I'm really working to pull together all of these different pieces of my research program to think more holistically about how forest systems will adapt to climate change from the ground up. And in Western science, we often think of trees as individuals and forests as sort of collections of individual trees. However, my research is really highlighting how microbes are actually critical parts of what are actually forest systems. And this is something that's been known in indigenous science for thousands of years. As my research program continues to investigate forest microbiomes and climate change, I'm especially thinking about the ways that my work can complement and amplify indigenous science around this topic. The Karuk Climate Adaptation Plan is an excellent example of indigenous science where microbial communities are thought of as parts of forest ecosystems, and they are explicitly included in their climate adaptation strategies. For example, they have found for a long time that this ectomycorrhizal fungus is an indicator species of forest health. Acknowledging my positionality as a white woman working in an academic institution, I reached out to the Karuk tribe to see if they would like their work highlighted in a review paper that I've been working on with colleagues at Stanford on how forest microbiomes respond to climate change. And I really wanted to highlight their work as an important example of indigenous sovereignty, especially with regard to climate adaptation. So while I'm the one that reached out to the Karuk tribe, I've made sure that our collaboration is indigenous led and they are co-authors on the publication that's now in revision. Our work together has really been an important lesson for me in how the compartmentalization of Western science can impede progress towards climate adaptation. Indigenous science is not an add-on to Western science. And by highlighting this work, my aim was to really emphasize the importance of indigenous sovereignty in climate adaptation. And I'm currently thinking of ways that I can more actively promote policies like rematriation or the returning of indigenous lands as a climate adaptation strategy. Complex environmental issues like climate change really require diverse perspectives, especially from communities most affected by the climate crisis. And I take my role as a scientist, mentor, teacher, and science communicator very seriously because I recognize that my work in these arenas will really help to determine whose voices are represented in future scientific spheres, including in who's involved in decision-making around climate policy. Forests are critical ecosystems for our planet, and ultimately, my goal is to create a collaborative research and learning environment to really help shape climate adaptation in an inclusive and equitable way. And with that, I'll take any questions.
Thanks so much, Claire. That was wonderful. Sometimes we get questions in the chat. Yeah, Amy's clapping. Everyone can clap actually audibly or using those emojis. But if you have questions, feel free to unmute yourself. We've been going through and muting everyone just during the talk, but feel free to unmute yourself and ask. Or it looks like there's a question on the chat now from Steve Singer. How well do mycorrhizae survive severe forest fires?
That's a great question. And I've actually been able to work on a project looking at the response of specifically redwood communities to forest fire with a graduate student and undergraduate student in my current postdoc lab. And what we've been finding is that some fungi are able to survive, but that there's actually quite a reduction in the overall soil fungal community, and a shift towards a more pathogenic community as well.
And there's another question. Do fires increase the viability of redwood seeds?
Also a great question, and something I've been working on with an undergraduate student. And we've been finding some really interesting results here. And what we've seen is that the forest microbial community post-fire, especially in medium and high severity burns, does increase redwood seed germination, and that even just the soil chemistry post-fire actually also helps to increase redwood seed germination post-fire. And we don't see that same result with Douglas fir. So actually Douglas fir seedlings do best in unburned or low burn severity, and they don't do as well post-fire in high severity burns.
So a question from Edie and Mike. Hello. So in your experiments, your competition experiments, you did inoculation studies, but how do you keep the ectomycorrhizal plants from being contaminated? So what are your experimental conditions? It's pretty easy to do for arbuscular mycorrhizal plants to inoculate them, but how did you do that for EMFs?
Yeah, that's a great question. So I was specifically using Pseudomonas pungens, which has been shown to be a really important ectomycorrhizal species in this specific environment. And unlike other types of ectomycorrhizal fungi, it doesn't travel around so easily. So there are things like Phyllepharas that are ectomycorrhizal that are easily wind dispersed, and those can have a ton of contamination. But this specific fungus that we used really didn't actually travel between pots. And so I checked colonization also in each of these treatments, and we didn't get any sort of contamination in the sterile or the non-mycorrhizal, not sterile treatments.
And so this was a greenhouse or a growth chamber study?
This was a growth chamber study. Mm-hmm.
Oh, okay. Then, all right. Very good. Thank you.
Yeah.
Can I ask another question? This is Mike Allen.
Yeah.
Great talk. Really nice. When we worked on pinyon juniper systems with fire, one of the things that we observed is when fire would come through, that you had a stand, here's a juniper and here's a pinyon, is that the recovery would reverse itself after the fire. And I'm just kind of curious why. I've always wondered, why would that pattern exist? We know that the surface communities are affected, surface fungal, and deeper they seem to be pretty tolerant, at least the AM fungi. So and I'm wondering in your redwoods, because it's so interspersed with ectomycorrhizal communities, here's some real interesting potential dynamics.
Yeah, I think redwood communities are particularly complex with fire, right? Because they can also resprout. So obviously seedling recruitment is going to be really, really important for genetic diversity and adaptation of these forests moving forward. But that's just one part of the story, and there is a lot of resprouting that does occur. I don't know as much about the pinyon juniper system, but I suspect that there's probably some host-specific like fungi or bacteria that exist post-fire that are probably driving a lot of those trends. But I don't know of any work specifically investigating that offhand.
Interesting.
I'll jump in.
Oh, go ahead.
Hi, Claire. Shauna Gofretti here.
Hi, Shauna. Thanks so much for coming.
Of course. Oh my gosh, you've accomplished so much since your time at Oxy. I was really drawn to your first study on microbial ecology and climate and the enrichment of actinobacteria around the roots and in low water conditions. And you made a point to say that they were filamentous. And so I was wondering, do you think that they play a similar role to fungi in enhancing soil aggregation or water retention? Or what do you think are they being helpful? Or are they just being recruited because of this positive selection?
I think it's probably multiple things. So Streptomyces was the particular group that became extra, or it was the most dominant in these dry soils. And Streptomyces has been shown to improve plant growth during conditions of water deficit. So I suspect that there's some recruitment of those fungi by the plants themselves. There's probably some interplay happening there, but also just that these filamentous growth forms kind of aid the bacteria themselves in accessing pools of water between the soil aggregates as the soil profile dries out. So while things like Bradyrhizobium, which was typically very dominant in the wetter soils, is unable to move around and then access important water nutrient or water resources, these filamentous bacteria are able to grow towards those and kind of survive essentially these drier conditions in soils.
Oh yeah, super fascinating. Thanks. And I love your paper on the redwood nodules. It's just amazing, amazing study.
Thank you.
I had another question on those nodules. That's really exciting because it's always been hard to figure out what's going on with redwoods. But one of the things that we look for in trying to find where our AMs are located is the region of elongation, in which you have, you know, this just behind where the cells have, the plant cells are starting to elongate in their numbers. I'm wondering, what is the structure of those nodules from the plant side? And is that something that can help trigger the AMs? That was really exciting and new, very new stuff.
Yeah, that's a great question. So these nodules are different in structure from, say, like a root tip. There's no root cap on them. And so in that way, they're a little bit less lignified than these sort of more woody tissues around the rest of the redwood root system. And so I suspect that they're able to kind of allow things to get in a little bit easier than say that a root tip itself might be able to. And I also sort of suspect that there's probably some hormonal signaling that's being localized to these specific nodules. So there's, as I'm sure you're aware, complex signaling between plants and fungi using strigolactones. And I sort of hypothesize that there's maybe localized strigolactone production in these nodules that's allowing the mycorrhizae to essentially start that whole cascade process to colonize and elongate.
There's also a few questions coming in the chat that I don't want to overlook. So one question is, given the rate of climate change, people have been suggesting the need for assisted migration for plants. Do you foresee that this might be a potential for fungal communities?
Yeah, that's a great question. I've gotten that a lot lately. And I think that when we're talking about assisted migration as potentially like a climate adaptation strategy, my work's been really highlighting that we need to be thinking about the microbes. So doing assisted migration in the absence of microbes is probably going to be unsuccessful, right? And so, yeah, I think that when we're talking about climate adaptation and assisted migration, these fungi are also needing to be included in our views of how that's going to take place and if that's going to even be a viable strategy.
Very cool. Then there was a question about your animations. They are super cool. And so Beth Corbin asked about the ones drilling down to the different scales, especially within ecosystems. Did you find those animations or have them made?
The animations were from Cal Academy of Sciences. Yeah, so those are cited in there, but yeah, they're very well done, and I appreciate Cal Academy of Sciences for doing this.
Yeah, they're really cool. And then another question about, is there anyone collecting redwood seedlings from a variety of areas, specifically to repopulate burned areas? If you know.
I'm sure that there are lots of people doing this and lots of groups. I know that Cal Fire does lots of extensive redwood seed collection, for instance. So I know that these seed banks exist for potentially recovering redwoods, but I don't know of any explicit studies that are currently doing that at the moment.
And then this last one is more of a comment than a question, but I want to echo this also. Pleased to see that scientists are recognizing Indigenous communities and understand plant communities and all the complex relations better than we've given them credit for in the past, which is, anyway, I really appreciated your Indigenous science piece also. That was very exciting to see how that's coming together.
Would you have time for one more question? Sorry to butt in.
I could do one more quick question.
All right, sorry about that. I don't know if you can answer this or not, but maybe you can help. So I work in redwood forestry as a consulting botanist, mostly with rare plant population monitoring and counting and discovery and mitigation. And I come into situations where I'm trying to advise either a veg tech doing invasive plant maintenance, or spraying something, or doing mechanical removal of something like broom or jubata, or a forester trying to do a best practice measure while harvesting timber. And we've seen even now sequoia start to retreat, especially in the drought, to lower elevations, to floodplains, as to be expected. But I'm wondering if you have any ideas on best practice measures for maintaining soil health during disturbance events such as timber cutting. We're in a pro-silva company, we replant and everything, but me and my coworkers on the biology side kind of worry about the long-term health of soil during reforestation with plants that we think are especially sensitive, like some of the like the Piperia species and such.
Yeah, that's a really great question. And I haven't explicitly looked at any of this myself, but I do think that there's going to be some important potential like inoculation that you could do. So I don't think we're quite there yet with the research that I've done, but potentially including as you're replanting, for instance, arbuscular mycorrhizal fungi in those replantings, or potentially some of these bacterial species as well, could be really, really important in kind of making sure that as we've removed some of those trees, that those communities of microbes can still exist and help them to sort of reestablish in those systems. But in terms of where we're at with the research, I don't think we're quite there yet. And I can't just say like, oh yeah, it's this species and that's one you add, or like this conglomerate of species and that needs to be there for them to be successful. But I'm hoping to be able to get to that point in hopefully the next 5 or so years.
It's very needed. I look forward to it if you ever into it.
Awesome.
Well, it's past 8 o'clock, so I'm going to say thank you so much to Claire. That was amazing to hear about. And I know a lot about your research, and so I learned a lot of new things today, which is really exciting. So thank you all for being here. And then we'll see you hopefully in June for our next speaker. I'll have to look it up. I think his name is Avery. So we'll see you then. Take care.
Thanks all.
Thanks so much.